Hello everyone, welcome back for more proteomics. So this is going to be a fairly short set of lecture videos. I wouldn't really anticipate any of these videos going much longer than 10 minutes. But this is going to cover a fairly important subject matter of which we're going to become aware that not every protein is fully soluble in a bodily fluid or in solution. So some proteins are actually embedded within membranes, whether it's the plasma membrane or the membranes of intracellular organelles. And this is going to be important because it's going to allow us to apply a lot of some of the introductory principles of proteins that we've talked about before, especially those regarding how amino acid side chains interact with their environment. So without further ado, let's start off by reviewing a little bit about what we already know about the cell membrane and membrane proteins. So we've already had discussions in class before about the cell membrane, which of course is composed of a bilayer of phospholipids. So we've talked before about why the cell membrane arranges itself like this, so we don't need to rehash that topic. But we also discussed that embedded within the membrane are going to be a wide variety of different kinds of membrane proteins. Now, these membrane proteins are going to serve a lot of very diverse different functions inside the cell. A lot of them don't necessarily have a whole lot in common with each other than the fact that they do happen to be in the membrane. So we'll try to provide as many examples of them as we possibly can. So we've talked about this before in a previous lecture, but these are just some of the classes of membrane proteins that you may encounter, some of what you can expect some of these proteins to do. We've talked about channel and carrier proteins, which can function in membrane transport, allowing molecules and other materials that would otherwise be unable to cross the cell membrane all by themselves to do so by moving through a pore that is formed by the channel or by the carrier. We've talked about cell recognition proteins, which are typically going to have attached carbohydrate chains on their extracellular fluid facing side. We'll talk about some of those a little bit later on, but we've talked about how these can be involved in a lot of cell recognition machinery. Receptor proteins we haven't really talked a whole lot about, but we will be looking at a specific example of a receptor protein a little bit later on. Enzymes we've talked a lot about before. Obviously, with tyrosinase, we are studying this semester in lab, we're studying an enzyme, but tyrosinase is actually a soluble enzyme, meaning it's not actually embedded within the membrane. But we would be remiss without mentioning that absolutely enzymes can be embedded within the membrane, and we will eventually see some examples of that too. And then finally, junction proteins are structural elements that are embedded within two different membranes, and these help to hold cells within tissues together. So as far as membrane proteins go, there are two major varieties. There are integral membrane proteins, the type that tend to span the entire width of the membrane and are actually anchored within the interior of the phospholipid bilayer. So these typically are going to include your channel proteins, your carrier proteins, junction proteins, and then also some types of receptor proteins. And then on the other hand, you have peripheral membrane proteins, which are subtly anchored to one side of the membrane. So you may find one of these proteins attached to either the extracellular fluid side or the intracellular fluid side, but they are quite clearly interacting with just one and not the other. And they are usually interacting with that side of the membrane not through a permanent attachment to the interior of the cell membrane, but rather by either attaching or glomming themselves onto another type of protein that's already a part of the membrane, or by having a lipid anchor, which we will talk about at the end of this lecture. So let's focus on integral membrane proteins for now. What sets them all apart is their ability to be anchored in the cell membrane through what are called transmembrane domains. A TM domain refers to a stretch of amino acid residues, most of which are going to be hydrophobic, And what's going to happen is that this transmembrane domain will insert itself into the phospholipid bilayer such that these hydrophobic side chains are capable of interacting with the fatty acid tails of the phospholipid bilayer. So if you look at this picture over here in which we have a transmembrane domain that you can clearly see is inserted into the phospholipid bilayer, if you look at the single letter codes of these amino acids, you're going to see a lot of hydrophobic ones. So just kind of giving it a brief glance, you can see phenylalanine, alanine, alanine, leucine, valine, valine, leucine, leucine, glycine, phenylalanine, leucine, alanine, valine. So you see quite a few hydrophobic amino acids. And the major principle that we've talked about before that underlies how proteins fold and how amino acid side chains interact with each other, that same principle still absolutely applies here. Hydrophilic amino acids like to interact with water, and they like to interact with other polar substances. Hydrophobic side chains, similarly, want to stay away from water, and they want to interact with other hydrophobic molecules. And by doing so like this, the side chains that make up the transmembrane domain are able to interact with other hydrophobic substances, that being the fatty acid tails of each phospholipid, and in doing so they also achieve their primary purpose, which is to stay away from water. So a single membrane protein may have just one transmembrane domain that goes through the membrane once, or some of them may have several of them, in which case the protein will appear to wind in and out and in and out of the membrane several times, one for each transmembrane domain. An example of one of these that we will look at later are called Gproteincoupled receptors. We'll look at them later, but they, as a shining example, actually have 7 transmembrane domains, meaning that the protein winds in and out of the membrane a total of 7 times. So here's the Gproteincoupled receptor that I just mentioned. These are going to be a type of receptor protein. Remember, that's one of the categories of membrane proteins that we mentioned. These are very commonly involved in cell signaling pathways, especially when it comes to mammalian physiology. So if you look at these, you should note how there are a number of different kinds of Gprotein-coupled receptors here. The ones we're looking at happen to be those that are either sensitive to light or sensitive to membrane potential changes. But what you'll notice that they all have in common is that they all have 7 transmembrane domains, and you can see they're all numbered here, 1, 2, 3, 4, 5, 6, 7. And as you can obviously see that I mentioned before, the protein itself winds in and out and in and out of the membrane a total of 7 times. And it just so happens that this sort of 7 passes through the membrane characterization, this just happens to be kind of a thing with Gprotein-coupled receptors. It just happens to be something that they all kind of have in common with each other. So a lot of membrane proteins, including both peripheral and integral membrane proteins, they may have carbohydrate chains that are anchored to the extracellular fluid side of the membrane. Now, it is important to note, and this is very important for you to remember, these carbohydrate chains will only be found on the extracellular fluid side. You will not ever see carbohydrate chains sticking inward towards the cytoplasm of the cell. So, like we said, these sugar chains are generally going to be a part of cell recognition profiles, basically giving your cells a fingerprint so that your immune system can recognize them, and this forms the basis for things like blood type and other sorts of things. Another thing that we did not get a chance to mention about the cell membrane last time is the importance of cholesterol, which is present in the hydrophobic core of the cell membrane. So every now and then you will find a cholesterol molecule, which is a steroid type of lipid. These steroid molecules like to wedge themselves in between the phospholipid tails, and they stabilize themselves by forming hydrogen bonds with the fatty acid carbonyl oxygens, and you can see that hydrogen bond here. Otherwise, the most hydrophobic portion of the cholesterol molecule is free to interact with the fatty acid tails, which are of course very hydrophobic as well. Now, the purpose of cholesterol doing this is that By having cholesterol present in the cell membrane, it spaces out the fatty acid tails of the phospholipids, allows the whole phospholipid bilayer to take on a much more fluid characterization so that we don't end up with cell membranes that are more solid, which is what you would expect with highly dense fats packing together, like what you see with butter and lard. Because the cholesterol spaces these things out, the membrane can take on a much more fluid nature, and it has a much lower melting temperature, which at a body temperature of 37 degrees Celsius, it still ends up being a fluid, which is what we want. And as I was kind of alluding to, the more solid the membrane is, the less efficiently a lot of those membrane transport processes are going to be, and we certainly do not want that. All right, so that's going to do it for this first video. Thanks for listening, and join us next time, and we will dive a little bit deeper into some of the secondary structural elements underlying the transmembrane domain by talking about the alpha helices that are hydrophobic. So thanks again, and I will see you next time. So long.